Hey everybody, Mr. Ling here. I just wanted to go through this one lesson really quickly with you, Heredity Patterns 3.04, as it is talking about some things that might be a little confusing. So I just wanted to go over, especially Punnett squares and how that works. But a couple things to note here. Heredity patterns, basically what you're doing in heredity patterns is discovering the probability of a certain genetic outcome. So if you cross two different species, or not two different species, if you cross two different individuals, hopefully within the same species, and you produce offspring, what are those offspring going to look like? We have to know a little bit about what traits are dominant and what traits are recessive. But your probability, depending on how what we've learned now about meiosis and how chromosomes are copied and split up and shared into four different daughter cells, the probability of those daughter cells coming together and producing offspring, and what kind of offspring they would make. Okay, so some genetic vocabulary that we need to know here. First of all, when we're talking about um, genetics and we look at traits, we are talking about a certain site on a chromosome called the gene, and that gene is responsible for certain traits. So, in one case, this example here talks about flower color. So, of these pea pod plants here. So, the large P is a purple flower, and the small p stands for a white flower. And the reason they use the uppercase and lowercase is that the uppercase always stands for the dominant trait, and the lowercase always stands for the recessive trait. And I'm sure you've read up on the dominance issue, but basically, if you have the dominant allele for a certain gene, then you only have to have one of those to express that trait. Now, the reason we have two, well, think about it. What we learned about back in mitosis is, in meiosis, I guess it was, is the number of chromosomes that we have. We have a double set. So if we're talking about really any organism, you have a double set of chromosomes. So you have, for instance, humans. We have 23 chromosomes. In other words, we have 23 different chromosomes that all have codes for different things that are important to life. Mostly our DNA produces proteins. That's what's made from DNA. But we have 46 total because we have one set from mom and one set from dad. We have that diploid number, if you remember that. So when those diploid cells go through meiosis, they split apart. They make a copy of themselves. They split apart, and they split into four different daughter cells. So two of those daughter cells have mostly dad's information, and two of those daughter cells have mostly mom's information. But remember, those because of crossing over and all that, they share some things too. But anyway, the bottom line here is we get four different daughter cells, and we end up having about a 50-50 chance there of getting mom's or dad's information. So, big P's and little p's, that's what we're looking at here. That's the two big P's together. That means you're double dominant, or you have both dominant alleles. Then that would be you're to be homozygous. And if you've got one of each, so if you've got a big P and a little p here, or one dominant allele and one recessive allele, then you're heterozygous, meaning kind of a mix. Homo means the same, hetero means a mixture. Okay? Now, phenotypes and genotypes. So phenotypes, pH, are the traits that you see. So in other words, in the flowers, we would see the purple flower. We would see the color. That's what the genes would give us. The genotype is basically what's on the inside, the genetics, the DNA. That's telling our bodies which trait to make, right? So we don't see what's on the inside. We don't see that DNA. We only see what it produces on the outside. So genotypes, what's on the inside, what's in your genes. So a good way to remember it. And a phenotype is what you look like on the outside. Okay? So there is that. And again, if you've got the heterozygous phenotype, you're going to show the dominant trait because you only have to have one dominant gene. That's why it's, or one dominant allele. That's why it's called dominant, because it dominates over the other one. The only way you're going to show the recessive trait is if you have one of each from each parent. You have a recessive allele from each parent for that trait. Okay? All right, so this gets into the idea of Punnett squares, and this is what I really wanted to talk about, is how these work. So again, these two S's here, when these, whatever trait this is, I don't know what it is here, but in this case you have a cross between, this is one individual, say dad, and this one's mom here, okay? And they're both heterozygous, meaning they both have one dominant and one recessive allele for whatever trait this is. So when we set up our squares, we split these up, because remember I said when a cell divides, you've got about a 50-50 chance of getting, the daughter cells, you're going to get two daughter cells that have the dominant sides and two that have the recessive sides in this case. If these were both big S's, then you'd obviously have 100%. But you still have to split them up like this so we can see how they're going to match up with the other side, okay? So we just basically split those into mom and dad, or I'm sorry, not mom and dad. This is mom, this is dad. We split them into the two different alleles. And they might be the same, dominant or recessive, or they might be different, like in this case here. So, and this just shows you what goes on to happen when those get crossed, the different combinations that they would make, how they match up. So this square here would hold this cross and this cross, which is the two big S's here. This square here holds this little s and this big S. So you just match them up, in line there. And you get these 4 different outcomes. You get one possible outcome is 2 big S's. One possible outcome is 2 little s's, and then 2 outcomes here are a big S and a little s. And again, the color just shows these came from mom, these came from dad, or whichever case that was there. So if you look at, this is again probability, right? This is saying that you got, for genotypes anyway, again, this is what's on the inside. For genotypes, you have about a 50-50 chance of getting a heterozygous individual from this certain type of cross. You've got a 25% chance of getting a homogeneous dominant, meaning 2 dominant alleles, and you've got a 25% chance of getting a double recessive or a homozygous recessive allele. And again, there's 4 squares, so basically that's 1 out of 4 or 25 out of 100 if you want to go by percentage, so 25% for each square, and that's where we get the probability from. Now, what I want to talk about is what happens when you get let's see on the next page here. What happens when you get 2 different traits that you want to look at? So you get, and again, these things get independently sorted. That's what this talks about up here. When you're crossing things, these are going to mix up in different ways, and I wanted to show you how that works exactly. So let's go to my board over here so we can see. I think this makes it a little bit easier. So in this case, we were going to cross, let me look again, it is 2, these are both big R, little y, big Y, little y, big R, little y, big Y, little y. These are both heterozygous for both traits.